Hello, and welcome to our first video for Unit 0, and we are going to be talking about Chapter 2, which is all about chemistry and macromolecules. The first section is just chemistry, so let's get right into it. Yes, we are going into chemistry again, just for a little bit. It is very important for anatomy and physiology. Anatomy is associated with the structures. Physiology is associated with the function of those structures. And both of these rely on chemical reactions and chemical processes to be built, if we're talking about structures, and to happen, if we're talking about functions. Okay, well, very basic stuff. Matter is anything that has weight and takes up space. It is just anything that is composed of elements being any of these unique atoms that we have here. Okay. An atom is the smallest complete unit of an element. The atom is going to include a nucleus at the center, which has neutrons, which are not charged, and protons, which are charged. They are positively charged. So proton P, positive P. And then we have electrons that are going to be in a cloud around the nucleus. Okay, electrons are smaller than neutrons and protons. They have the same size of charge as protons, but they are negatively charged instead of being positively charged. An atom is going to have the same number of electrons as protons, meaning that it will not have an overall charge. An isotope is going to be an atom of an element that has a different number of neutrons than a different isotope of the same element. Okay, so we're talking about two different atoms of the same element that are different by having different numbers of neutrons in their nucleus. So they'll have the same number of protons and electrons, so they'll be the same element. However, they'll have different neutrons, so they will have more or less mass because of that. Isotopes can release radiation depending on how unstable they are. You have alpha radiation, beta radiation, and gamma. Alpha, these are going to be big ones. It's 2 protons, 2 neutrons. Beta is going to be 1 electron, and gamma is going to be about what an X-ray is. So this is going to be lots of energy, still some energy, but less, and then even less here. Okay. An ion is going to be an atom that has either lost 1 or more electrons or gained 1 or more electrons. And in doing so, it will have a different number of protons and electrons and will have a charge. If it loses electrons, it'll be positively charged. If it gains electrons, it'll be negatively charged. Okay, ions are very important for a lot of different functions in the body. And the two that we're going to talk about most significantly are going to be in the nervous system for the propagation of an action potential, and also in the muscle for muscle contractions. Ionic bonds are when you have 2 oppositely charged ions, so 1 negative, 1 positive, and they are attracted to each other because they are opposites. So they will form a very strong attraction and, essentially stick together. A covalent bond is when 2 different atoms share electrons in order to complete their orbitals, which is what atoms want to do. They want to have a complete set of electrons in their orbitals. And to do so, they will either gain or release electrons like in an ionic bond, or they will share them like we have here in a covalent bond. And they can form single, double, or triple bonds depending on how many electrons are being shared. A molecule is a particle that contains 2 or more atoms that are covalently bonded together. A compound is going to be a molecule that has 2 or more different atoms. Okay, we'll talk about molecules later that are called macromolecules that have just a lot of different atoms. Sometimes when you have a covalent bond and the electrons are being shared, they're shared unevenly. And when that happens, we'll have one end that is positively charged, one end that is negatively charged. So in the case of water, the oxygen really likes the electrons. It will hold the electrons closer to it and will be negative. the hydrogens will have the electrons being held farther away from them, and they'll be positively charged. So as a result, we'll have a negative charge here, positive charge here. And when you have two different two different water molecules, they'll form what are called hydrogen bonds or just polar attractions, where the negative end on one will be attracted to the positive end of the other. So this is essentially like a very weak ionic bond. Hydrogen bonds are especially strong polar attractions. So this is whenever you have hydrogen bonding with either oxygen, fluorine, or nitrogen. And in this case, what we're going to talk about most often are water and how they form hydrogen bonds. Okay, if we have chemical reactions, a synthesis reaction is when we have two smaller atoms or molecules that are going to bond together and form a larger molecule. A decomposition is going to be the opposite, where we have a larger molecule and it breaks apart into two smaller molecules or atoms. An exchange reaction, we have two different molecules that are just essentially changing parts of themselves, okay? So if we have A, B, and C, D, then B and D switch sides, and now we have A, D, and C, B. A reversible reaction is going to be a reaction where, yes, it's going to go forward like what we saw before, but it can just as easily go backwards. An acid is going to be a substance that releases hydrogen ions into water. So basically, whenever this hydrochloric acid is put into water, it breaks apart, releases the H plus. So it's going to produce a solution that has lots of H plus in it to be an acid. A base is going to be opposite. It's going to be something that takes the H plus out of the solution. So it's something that will react with H plus and will lower the amount of H plus in the solution. pH is going to be a measurement of the hydrogen ion concentration in the solution. So this H plus, the larger the amount of H plus in the solution, the higher the pH will be. It's a logarithmic scale. So if we have 6.0 and 7.0, that means that to go from here to here, this is 10 times less acidic. Right, so the more acidic things are going to have a lower pH. The less acidic things are going to have the higher pH. This is going to be the basic side. This is going to be the acidic side. And the lower the pH, the more acidic it is. The higher the pH, the more basic it is. And so it is a logarithmic scale, meaning that for every one difference in pH, you will take 10 to that power. So if it's a difference of 1, it's 10 to 1, which will be 10. So that means that this is 10 times more acidic. All right. But what about 7 to 5? Well, that's a difference of 2. So you take 10 to the power of 2. That's 100, meaning that this is going to be 100 times more acidic than this. All right. And by extension, going down to 4. All right, 7 minus 4 is 3. So that means that something with a pH of 4 will be 10 to the third or 1,000 times more acidic than something with a pH of 7. Okay, now let's get on to the organic macromolecules. So if something is organic, that means that it contains both carbon and hydrogen. Make sure you remember that. You're going to see that question many, many times. Organic molecules are usually a lot larger than inorganic molecules. That's why we're kind of calling them macromolecules, the very, very large ones. An inorganic molecule is not going to contain both carbon and hydrogen. Maybe it contains carbon, maybe it contains hydrogen, but not both. All right, so some inorganic molecules that are important for us are going to be water, oxygen, carbon dioxide, and salts such as like calcium phosphate, which is what makes our bones rigid. So the 4 different organic macromolecules are carbohydrates, lipids, proteins, and nucleic acids. And each of these macromolecules are built through bonds between smaller parts, which we're going to call monomer units. So basically, they're just a series of repeated monomer units. And so a monomer unit is one of the small, essentially a puzzle piece. A polymer is going to be like the whole puzzle put together, all those pieces being bonded together. All right, so some examples of polymers for carbohydrate. For us, glycogen. Glycogen is made up of many glucose, which are going to be having a really important role in cellular respiration. Lipids, like a triglyceride, is going to be part of a glycerol and 3 fatty acid chains. A protein is made up of many amino acids. A nucleic acid, like DNA, is going to be made up of many nucleotides. Okay, so we're going to start off with carbohydrates. Carbohydrates contain carbon, hydrogen, and oxygen. They typically have the ratio of 1 carbon to 2 hydrogens to 1 oxygen. They are polar, and that means that they will be soluble in water because water is also polar. The monomer of carbohydrate is going to be called a monosaccharide. So mono meaning one, a saccharide referring to a sugar. It's one ring like what we have here. A dimer is going to be 2 rings bonded together. So a disaccharide, so di being 2, saccharide associated with a sugar. Polymer is going to be more than 10 rings, aka a polysaccharide. So monosaccharides, some examples that we have are glucose. That's going to be the big one that we use, but also fructose and galactose. Both of these have relevance. You'll see fructose in a lot of artificial sugars. Sorry, not artificial sugars, but in a lot of like sodas and juices. Galactose is part of lactose. And then we also have deoxyribose and ribose. These are the 2 sugars that are part of nucleic acids, deoxyribose being DNA, ribose being RNA. Over here, we have disaccharides, which are just a combination of 2 monosaccharides bonded together. Sucrose is glucose and fructose. Lactose is galactose and glucose. Maltose is glucose and glucose. Polysaccharides are going to be the big, long chains or huge networks of monosaccharides bonded together. Glycogen, I have it starred here. That's what we are going to be referring to most because that's what we store sugars as in our cells. Cellulose is going to be part of a lot of plants. It's part of cell walls. Starches are going to be part of, things like fruits or potatoes. Different types of monosaccharides: glucose, fructose, galactose, and deoxyribose and ribose. Do you need to know each of them individually? No, but should you know the general structure of a monosaccharide? Yeah, absolutely. Okay, so they have this ring structure. Each of these points of these, unless indicated otherwise, like this O here, these are carbons, and then they have either the hydrogens or the oxygen and the hydrogen coming off of them, all right? So glycogen, these are glucose stored within our cells, just a very large network of those. Cellulose, these are the rigid cell walls, so you can actually see it in the celery here. Starches are easier to digest for humans. They're in things like potatoes. And then chitin, which is less common, but a lot of times it's going to be part of exoskeletons of certain bugs. What do we use carbohydrates for? Well, we use glucose as quick energy. We can store it as long-term energy through glycogen, this is it right here. But they also provide the raw materials for certain cell structures, such as these little, essentially name tags here, these glycoproteins or lipids in the cell membrane. Moving on to lipids, so our second of the macromolecules. Lipids are nonpolar. That is really the characteristic that they all share. So they also contain carbon, hydrogen, and oxygen, but less oxygens than carbohydrates. And phospholipids are going to contain phosphorus. That's what this P is. So these are fatty acids here, just chains of carbons and hydrogens. An unsaturated fatty acid is going to be one that has less hydrogens than a saturated one because of a double or triple bond. Okay. So a triglyceride is going to have 3 fatty acids attached to a glycerol backbone that we have here. A phospholipid is going to be just like a triglyceride, but only 2 fatty acids. And instead of the third one, it has a phosphate group. Steroid is going to be this 4-ring structure where they share carbons. and they can have a whole bunch of other things coming off of them, but this is just the general structure of a steroid. So an example of this would be like a cholesterol. All right, triglycerides, again, 3 fatty acids, these long carbon-hydrogen chains. The glycerol is going to be this carbon-hydrogen-oxygen backbone, and they help to store energy. I mentioned before, fatty acids, if they're saturated, that means that they have all single bonds. They have the maximum amount of hydrogens. You typically will find saturated fatty acids in animal meat or dairy, in addition to certain oils. And unsaturated fatty acids, these are generally considered to be the healthier ones, are going to be found in plant fats like oils and nuts. This is a diagram of the glycerol, so the glycerol backbone, and this is where the fatty acids will bond to. Phospholipid, again, is just like a triglyceride or a fat, but instead of the third fatty acid, it has the phosphate group. The phosphate group is polar, unlike the fatty acids in the glycerol. So that means that the phospholipids are going to have a non-polar region and a polar region. This is going to be really important for the cell membrane that is made up of the phospholipids. Steroids are going to be these 4 ring structures that can have these other parts connected or bonded to them. And one of the most important functions of a steroid is going to be to provide structure within the cell membrane to prevent too much movement, but they're also used as hormones as well. Okay, that brings us to proteins. So proteins are made up of carbon, hydrogen, and oxygen, just like the other 2. Also nitrogen, also sulfur, and some. So it depends on the amino acid, but all of them are going to have carbon, hydrogen, oxygen, and nitrogen. So their monomer units are called amino acids. They contain a carbon in the middle, a amine group on one side, a carboxyl group on the other. So NH2, the amine, COOH, the carboxyl group. And the side chain is going to be one of 20 different things here attached to the carbon as well. And when we have 2 amino acids coming together to form what is called a peptide bond, basically we'll see a bond between the amine group of one and the carboxyl group of the other. This is the distribution of all the different amino acids. Do you need to know all these? Well, for biology, you would. Maybe for a future anatomy class, you would, but you're not going to need to know them for here. This is just for your reference. So you can see all the crazy R groups that we have on these different ones, some as big as this for tryptophan. All right, but it is important too for the structure because some are polar, some are non-polar, some are charged, some are not. Well, both of these are charged, but they have different types of charges. Okay, proteins are going to form from a chain of amino acids. So the primary structure of a protein is just going to be that order of the amino acids. The secondary structure is when they start to interact together to form hydrogen bonds. The tertiary structure is when we have more hydrogen and covalent bonds. And a quaternary structure is when we actually have multiple strands of polypeptides coming together. Okay, so basically what I just explained there. So the sequence of the amino acids for primary, when we start getting hydrogen bonding, we get the beta pleated sheet, like here, the alpha helix, like we have there, through hydrogen bonding, through the oppositely charged or polar parts of the amino acids. The tertiary is when it folds even more. We get that really complex structure of both the beta pleated sheets and the alpha helix, and even more just changes in the shape. Quaternary structure is when they different polypeptides come together. Okay, so not all proteins are going to have this quaternary structure. There are a lot of different functions for proteins. We use it as an energy source, kind of as a last chance, after using all the carbohydrates and fats. Use it as structural material, use it to deliver messages as hormones, use it to receive chemical messages in the within cells or within the cell membranes. use them to fight pathogens as antibodies, speed up chemical reactions as enzymes, interact for muscle contraction as actin and myosin, transport oxygen, that's hemoglobin, right? So based off of whatever the protein is, there are a lot of different functions here. All right, so we got keratin here, big component of hair. We've got the receptor within the membrane or even a receptor within the cell. We could also have a hormone that is made up as a protein. So we got different examples there. Antibodies have this kind of Y shape, but they are made up of, they are a protein. Enzymes are also a protein. They help with chemical reactions by either splitting something up or bringing them together to form a bond. Muscle contraction, we're going to learn a whole lot more about this actin here and this myosin here and how they interact to help contract a muscle. Hemoglobin, this is a quaternary structure here because there are beta globin and alpha globin here that come together to form the larger structure of hemoglobin. They transport oxygen. All right, and then finally, that brings us to nucleic acids, and it's made up of carbon, hydrogen, oxygen, and nitrogen, as well as phosphorus. So it has all 5 of these. Nucleotides are the monomer units. They're made up of a sugar in the middle, a phosphate off to the side, and a base really towards the center. A nitrogenous base. The polymers are going to be chains of these nucleotides, which are going to be either DNA if it's double or RNA if it's single. A nucleotide is going to be composed of the sugar, either deoxyribose or ribose, the phosphate group, which is PO3 minus, and the nitrogenous base, which is one of 4, depending on if we're talking about DNA or RNA. So for DNA, we have thymine, adenine, cytosine, and guanine. And for RNA, we're actually going to substitute thymine for uracil, which I'll talk about in a moment. But the cool thing about the nitrogenous bases is that they are complementary, meaning that this thymine will always bind with adenine. This cytosine is always going to bind with guanine, which helps with certain things like DNA replication, as well as transcription and then translation later. So DNA, its structure, we got 2 strands. So we're going to see the sugar and the phosphate group bond together. That forms one backbone over here. And we've got the phosphate sugar backbone over here, and this one is flipped. And between these 2 strands, the opposite bases, whether this is adenine and thymine or guanine and cytosine, they're going to form hydrogen bonds to stick together. So hydrogen bonds are not covalent, they're weaker, they will hold the DNA together, but it will be easy enough to break it apart whenever we need to do something like express a gene. All right, RNA is going to be just a single strand. It's going to be a lot shorter because it's really just a copy of one gene, which a DNA molecule will have many genes. So it's a lot shorter. It will substitute uracil for thymine. And that's pretty much it. So RNA, while DNA is going to help to store genetic information, RNA is going to copy it down. This is essentially like going into a book and taking a note out of it. But it will take the genetic information out of the nucleus, go to a ribosome, and that is where it will be used to synthesize a protein. Okay, so this is the whole point of nucleic acids, to go from our library of information to express the genes by producing proteins. Okay, so transcription is where we make a copy of the gene, and translation is where we translate it from nucleic acid or nucleotides to amino acids that will build the protein. Okay, and our final example of a nucleic acid is ATP. We use this for energy. This is going to be the final product of our cellular respiration, and we're going to use this for energy. So basically, ATP is the same as a adenine or a nucleic acid with adenine on it, but instead of only having one phosphate group, it has 3. And to access this energy, we're just going to kind of split it here and pop off that last phosphate, which will help to release energy that we can use for a chemical reaction. Okay, so that is the end of chapter 2. I know this is rather long-winded, but I hope that you have found this helpful, and I will be back with chapter 3 in a little bit.